Welcome to this webinar. I'm Barbara de Koning, professor at the Crop Biotechnics division at KU Leuven, and with this presentation I would like to provide you with more information on how the CRISPR-Cas technology can serve as an important tool to support sustainable crop production. We're all aware that we have to evolve towards more sustainable agricultural systems with less impact on the biodiversity of our ecosystems. Both the growing population and the impact of climate change will make this a very difficult challenge. It will ask efforts from every stakeholder in the agricultural food chain and will require a better distribution of our food, less food waste, but we will also have to rethink our agricultural systems with an emphasis on the implementation of novel technologies such as precision farming and precision breeding. The latter will be necessary to develop climate resilient crops or crops with, for example, resistance against diseases so that less pesticides have to be used, or crops with increased nutritional value. We can partially achieve this via classical breeding technologies. Here you cross an elite variety in white that has a favorable trait with another variety that has a favorable trait, and you see the favorable trait in pink, for example, disease resistance. In order to end up with a variety that resembles as much as possible your elite variety, you have to backcross with your elite variety for several generations. Classical breeding is as such a very time-consuming and often becoming very difficult when your favorable trait is linked with a negative trait. Another technique that has been widely used is breeding through mutagenesis. Here you treat your elite variety with a chemical or radiation to induce mutations. Also, this is a time-consuming technique since you may have to screen many plants in order to retrieve the favorable trait that you're looking for. Moreover, not only your gene of interest will be mutated, but also other genes could be mutated. So both mentioned techniques, breeding techniques, are not always that specific. Since the start of agriculture, farmers have been selecting and breeding for optimal crop varieties. Here you see the famous examples of maize and carrot and how these varieties changed throughout the selection process. More recent examples, for example, are the apple variety Kanzi, which has been created in collaboration with KU Leuven, or, for example, the many tomato varieties that you can now buy in the supermarket. And as a last example, I would like to give you the pink grapefruit or the durum wheat. Those varieties were created through mutagenesis. A third option I would like to emphasize is the breeding through genetic engineering. Here we select the gene, the pink again, that encodes a specific trait and we transfer it to the elite variety in white, resulting in genetically modified crops. This gene can be derived from another species or from the same species, but another variety or from a wild cultivar. So here you have genetically modified crops with only one gene that's different from your elite variety. In this slide, I would like to provide you with some more information on how these genes are transferred to the plant. I'm not going to go into detail, but we therefore make use of a bacterium, Agrobacterium tumefaciens. This is a plant pathogen and causes disease by transferring part of its DNA to the plant. We use this characteristic of this pathogen and replace its DNA with our gene of interest, that it's coding for our favorable trait. Then we transform the agrobacterium back with this construct, and this agrobacterium we treat our plants with, so that the plants got infected and that actually our gene of interest is transformed into the plant. And like this, we have a GMO or a transgenic plant. Worldwide, there are at the moment 22 genetically modified crops grown, divided over 41 countries. And as you will notice, apart from Spain and Portugal, no GMOs are grown in Europe. And this is because of the very strict legislation. The most important GMO crops are, for example, soybean, cotton, maize and canola, and of course often grown in USA. And these crops are most of the time herbicide resistant, or they have resistance against insects, or they can even have both. So the discovery of CRISPR-Cas, so now I come back with the terminology of CRISPR-Cas, has also revolutionized genetic engineering of crops. Thanks to this tool, we can now very precisely edit the genome also of crops. And the toolbox of CRISPR-Cas editing technologies has been expanding very rapidly. We can now, for example, also specifically modify one base pair by linking a dead Cas9 that doesn't create a double-stranded break with, for example, a cytosine deaminase. As such, we can change a cytosine into a thymine. And after stable expression of our mutation, the CRISPR-Cas construct can actually be crossed out. And in the end, your product only contains the intended change in the DNA without any foreign DNA. And this mutation will not be distinguishable from natural mutations. And this is quite important as a message to take with. Here, I will not focus in this presentation on the European legislation, but I think it's quite clear from this map that a lot of countries don't see the GMO edited crops as GMOs. While in Europe there is still a huge debate going on, and at the moment they need to be regulated as GMOs. The many examples of genome-edited crops have clearly shown the huge potential of this technology. And here I'm providing you with only a few examples, but the list is much longer. And if you're interested, I would advise you to check the many examples that are present in the, for example, APHIS database. This is the database of the Animal and Plant Health Inspection Service of the USA. And there are also many publications available. So you see a link on this presentation where you can have a look at those publications as well. Now I would like to focus a little bit more on how we use genome editing through CRISPR-Cas within our division of crop biotechnics. We use it both for accelerating our fundamental research on model crops as well as other crops, as well as for generating, for example, climate resilient crops or disease resistant crops. So more applied research as well as fundamental research. And we work on several crops, for example, on banana, tomato, strawberry, apple, and our model plant Arabidopsis thaliana. So as long as it's a tasty plant, we work with it, apart from the Arabidopsis, of course. In the Van de Poel lab, lab within our division, they study the biosynthesis of ethylene, a very important hormone in plant aging and fruit ripening. And one of the enzymes they study is ACO, or aminocyclopropane carboxylic acid oxidase. This enzyme has 5 homologs in Arabidopsis, And the group has developed, so you see the 5 homologs here, this group has developed a construct with 5 single guide RNAs to knock out this complete family of ACO homologs. And as such, once you have this tool, once you have this plant with mutated ACOs, you can really perform your research on it. Okay, so in my lab, we engineer the engineer. What do I mean with this? So I told you about Agrobacterium tumefaciens. But this Agrobacterium tumefaciens also has a little brother called Agrobacterium rhizogenus. And this Agrobacterium rhizogenus causes a disease called hairy root disease. And hairy root disease, for example, occurs on tomato plants. Tomato plants grown on rock wool in our high-tech greenhouses. And as you will see, by 2016-17, we already had 45% of our greenhouses that were infected with this bacterium. And as the name says itself, hairy root or crazy root disease, we also call it, means that we produce, or that those plants produce way more tomato roots. And I told you before that Agrobacterium transfers part of its DNA into the plant. And here it does it as well. And on this DNA that it transfers it to the plant, there are several genes that encode proteins that lead to this hairy root phenotype. However, we don't know what these genes are exactly doing, and how they are doing this. And so this is part of the research we are doing in the lab. So we developed several systems to actually look at the role of those genes. So we inoculate our plants with this Agrobacterium and we see how they develop those roots. Of course, we also want to have mutants in those genes. But Agrobacterium is actually very difficult to mutate. And therefore, and now I come back to CRISPR-Cas, therefore we actually used the CRISPR-Cas technology. So we used a kind of base editing, because if you use the normal CRISPR-Cas with the double breaks, then it will actually kill your Agrobacterium, because Agrobacterium is a bacterium and cannot restore from these double-stranded breaks. So what we did is develop a system so that we don't make these double-stranded breaks, but that we can just edit specific nucleotides so that we introduce stop codons in our genes of interest. And when there is a stop codon, there will not be a protein, a functional protein we produced. So we did do this CRISPR-Cas technology, or this base editing technology, on several of those genes. And then maybe it's a little bit small to see, but later on we checked our mutants on a carrot disc whether they could still produce hairy roots or not. And you could see that our K599 or wild type strain could still induce a high production of roots, while, for example, the role B mutant, very important in developing those hairy roots, so this Agrobacterium strain could not produce the hairy roots anymore. Okay, then I would like to end with one other example, and this is precision breeding for resistance against Fusarium wilt. At the moment, we are not yet in the lab doing this precision breeding for resistance against Fusarium wilt, but this is something that it's really needed, because Fusarium wilt, caused by Fusarium oxysporum cubense, tropical race 4, is a deadly disease for banana. And you see the symptoms on the slides. And since 2019, this disease has also come into Latin America. And in Latin America, we have the big monocultures of our Cavendish banana that's threatened, or I mean the Cavendish banana that we eat as a dessert banana most of the time. So we really need to find alternatives to treat this disease. And one of the alternatives is actually breeding or precision breeding. Here, for example, the lab of Professor van der Schuren and the lab of Professor Pauwels, they developed a CRISPR-Cas technology which was very useful in banana, which is not that easy to do. And here they had a proof of concept where they mutated a gene so that the plants are completely pale, so white, or gene where the plants are a little bit chlorotic, so a little bit less green, showing that this CRISPR-Cas technology mutating those specific genes is working. So now we have to find resistance or susceptibility genes against Fusarium tropical race 4 so that we can also generate transgenic plants. However, I do want to emphasize that it is very important that we spend effort in all this research and in generating transgenic plants, genome-edited plants for the future, but that this is not the only or will not be the only solution for a more sustainable crop production. So this was the end of my presentation. I hope that I could tell you a little bit more on the importance of genome editing for fundamental research, but definitely for applications later for sustainable crop production. Thank you very much.